Thanks very much, and thanks for the opportunity to speak today. And I'm conscious that I'm sort of embedded in a very pragmatic afternoon session, and so I guess what I'm going to talk about may very much look like it's coming from left field. But I guess I wanted to give you a sense of where basic research can potentially impact in the longer term on breast cancer care, and to give you a view of the prism through which we look at cancer. So there are already some important parallels between our institute and New Zealand. So we were established just over 100 years ago, and actually our first director was at Gallipoli on the way back to Australia, and actually got killed in Gallipoli, so never made it back to Australia. So he, with many of the Anzacs, suffered some 100 years ago. And one of the things that our institute is very interested in doing is trying to leverage off basic discovery to try to work out how it's going to impact positively for patient care. But that's not an easy task. So the work that's done by our group and many colleagues at the Institute is often very fundamental. And there's a huge chasm. It's been called the Valley of Death to actually get a basic discovery into the clinic. And so it's quite important to have clinician scientists, I guess, like myself, that can actually be involved in patient care and understand the complexities of how basic research happens and how hospitals function or dysfunction, depending on your view of the universe. So what I obviously want to focus on today is breast cancer. Many of you will empathise with this. I personally know how terrible it is to have a cold, but I've looked after women who are busy doing the stuff on the top. So one of the questions, of course, that we ask at a very basic level is, well, how does breast cancer actually happen? Because if you can get an understanding of that, then maybe you can either switch it off before it happens, or at least identify markers and targets that will treat breast cancer itself. So a lot of this will actually look familiar. These are sort of what epidemiologists have told us for many years. In fact, the first removal of the ovaries was done in 1896 in Scotland by a surgeon who thought that the ovaries must have something to do with the breasts, because if you took that out of cows, they would keep lactating. And so there have been long-standing understandings for breast cancer risk being a disease largely of women, a disease of age. Of course, family history can be potentially very important. BRCA1 and 2, which I'll talk about. And then a lot of reproductive factors which are code for female hormones, estrogen and progesterone, and other things which will look recognisable there. But that kind of paints the epidemiologic picture of why cancer might happen. What our group is interested in understanding is what are the cellular events and the genetic events that actually impact on the sorts of things you saw on the previous slide, and how does it actually happen at a cellular basis? How do female hormones play a role, and can you actually use these learnings to try to make a difference for patients with breast cancer? And so the view that Jane Visveda, who is my partner in crime for this research, and I took, is that we really want to understand how normal mammary gland or breast development happens, because if we can understand that, then you're not that many steps away from breast cancer. So actually, to get a breast cancer from normal breast tissue, you probably only need about 8 to 10 what are called driver mutations that make that cell become cancerous. And so in many ways, breast cancer could be viewed as a caricature of normal development that has gone awry. So if we can understand normal, maybe we can understand how cancer happens. And so what I'd like to show you in the next few slides is just some images of not only the complexities, but in a sense how beautiful breast development can be in terms of studying it. So most of the action, of course, happens after birth. It's one of the few organs in the body that develops postnatally. Most organs are formed in the body when a person is born. There's a bit of brain development, things that happen, but the breast really only kicks into action at about the time of puberty. So just before birth, there's something called the unlager, which is just a primitive precursor set of tissue in the nipple area, Now this is a mouse, and mice are very busy between 4 and 6 weeks of age. That's when puberty happens in a mouse. And you can see these are the ducts which grow out from this unlager by the time you're ready for puberty. But once puberty kicks in and the female hormones switch on, you get this massive expansion of the ductal tree through the pressures, principally of estrogen, driving that ductal outgrowth through the breast tissue. Anne Rios, who was in that previous image, actually pioneered a new way of looking in 3D at tissue without manipulation, and that's a couple of these images that I'm showing you here, where you can see that a piece of the gland, or in fact most of the glands just before puberty, and you can see these highly specialised structures, including these myepithelial cells. So she's been able to do some 3D imaging where you can take a duct from within the breast tissue, and it's the ducts which are important because they're the bits that can give rise to cancer. And you can see that they're really layered with 2 layers of cells. There's the inner blue layer here, which is called the luminal layer, and that is really important for letting milk flow through the ducts, but can also be milk producing in the more periphery. On the outside, you have the myepithelial cells, which can actually have contractile properties and help squeeze tissue. And yes, so it's working. So these are 2 images looking at those 2 separate layers, and you can see quite amazingly in this time-lapsed photography, if I can get it to work, how the myepithelial cells on the outside are actually almost squeezing the ducts, and that's to help, for the mature gland to let milk flow through. And these are the inner luminal cells, which are the cells which actually are can give rise to breast cancer. And you can see they're almost sensing the environment and talking to each other. So while we're all sitting here, that's all happening in our mammary glands, but not quite at that speed. And then there's even more that happens as the gland prepares for lactation. There's a whole outgrowth of cells that are derived from a pool of stem cells, which can give rise to the milk-producing units. And the colouring here is a little genetic trick that we've carried out, or rather Anne and Nyang have carried out, where they can actually indelibly label a stem cell with a different colour marker, and then all the progeny from that produce a cluster of cells which are derived from a stem cell within that part of the mammary tree. And this is how different it can become. This is a mouse now that's lactating, and you can see how different developmentally it's all become with these, it's like a tennis court net of the myepithelial cells around this milk-producing factory. And we've been doing similar imaging using human breast tissue, and you can see there's actually, in one sense, not that much difference between human and mouse tissue in terms of the anatomy, but most importantly for us, in terms of understanding the key signalling pathways. So a lot of the lessons we're learning in mice we can apply to human, and vice versa, although humans arguably have the greater need. So in terms of thinking about how all of this happens, there's this, as I hope I've sold to you, there's this huge growth of cells that's required to produce a milk factory. And necessarily from birth and puberty, and to be able to do this, particularly through multiple rounds of pregnancy, you've got to be able to make lots of cells. And so stem cells are at the very heart of this process, because they're like the seeds that can give rise to that whole ductal tree. And so having those and having them behave themselves is going to be very important to stop cancers from happening. And a few years ago now, just over a decade ago, Mark Shackleton and Francois Vaillant in our lab spent a lot of time trying to identify stem cells in mouse breast tissue, and we were able to do that after many years of trial and tribulations. And we're able to find that even a single stem cell can give rise to the whole ductal outgrowth in that mouse tree. And over the last decade or so, we've been able to paint a picture of how we think the stem cells ultimately give rise to the mature descendant cells, which formed part of those original images that I showed you. And the reason for doing that is not just academic. We want to understand whether these cells are the culprit cells which can give rise to the different types of cancer, like hormone receptor positive cancer, triple negative cancer, HER2 positive cancer, etc. And in fact, by looking at the molecular footprint of these cells, the molecular markers, what we were able to do was to discover that the stem cells themselves look most similar to very primitive cancers that are quite rare in humans, called metaplastic breast cancers or Claudin-Low tumours. The triple-negative cancers often have something called a basal-like phenotype, and what we believe, certainly in terms of their molecular pathways, they're probably produced from daughter cells, from these cells we call progenitor cells, very rapidly dividing cells which are dividing so that they can produce the more mature cells. And these are probably the culprits that give rise to most triple-negative breast cancers. Whereas the hormone receptor positive tumours, which are often called luminal tumours, they seem to arise from the more mature cells within this hierarchical tree. So another question we've asked is, well, how do hormones set all of this up? Obviously, you saw that image of what happened from puberty as the breast grows in response to estrogens. And one of the things that we've been able to decipher is that there seem to be some mature sensor cells in the breast, and they respond to the female hormone progesterone. And in response to progesterone, they produce a special protein called rank ligand, and then that rank ligand can actually talk to adjacent precursor cells, the primitive precursors, and switch on the whole system. And so as a result, you get an expansion and growth of breast tissue. So of course, this is potentially quite important, because if you can switch off rank ligand signaling, you might be able to switch off proliferation in the breast. And I'll come to why rank ligand is an important or interesting molecule shortly, in fact fairly soon. So one of the next questions we asked was, well, what about women who have a genetic predisposition to breast cancer, like BRCA1 mutation carriers, most made famous by Angelina Jolie, of course, who have a very high lifetime risk of developing breast cancers? Is it the stem cells which are the culprit cells that give rise to cancers in BRCA carriers? And this probably will be familiar to most of you, but there's an increase in breast and ovarian cancer risk for women with a faulty BRCA1 gene. And my little back of the envelope calculation coming over here was that about 85 BRCA1 tumours probably happen each year in New Zealand. And if you do a back of the envelope calculation, there are probably about 6,000 women in New Zealand who carry a BRCA1 mutation. So boutique rare gene, but obviously at a society level, potentially quite important. And often the tumours that arise in BRCA carriers are triple negative. Excuse me. So one of the things that we were able to show was that it's this daughter cell that appears to be where the defect lies in normal breast tissue with women with a faulty BRCA1 gene. So as a result of having a single faulty gene, there's an increased predisposition to breast growth and proliferation of this progenitor cell, which can ultimately, or which can give rise in some women to a triple negative breast cancer. And so the reason I want to come back to this rank ligand molecule is that what we discovered was that these culprit cells actually express rank on their self surface. So this is the signaling trigger for rank ligand to signal in a cell and to switch on that proliferation. So what we believe happens is that you can get this excessive rank signaling in this daughter culprit cell, and it can undergo cancerous changes and eventually, for some women, give rise to a breast cancer. And importantly, there are drugs in the clinic, which some of you will have heard of. There's a drug called denosumab, Xgeva is the other name that some women will be familiar with. It's actually used to treat osteoporosis or breast cancer that has spread to the bone. And so this is a potential opportunity here for repurposing this drug. So instead of using it to treat cancer that's gone to the bone, to actually look at the question as to whether or not we can prevent these prone cells to becoming cancerous before, so to either delay or prevent a breast cancer from happening. And so this has some important relevance for New Zealand because Emma Nolan from our lab is actually a New Zealander, so it's a bit like the Pavlova and Russell Crowe. You could argue that this is an Australian discovery. As a result of this work, what we've been very keen to do and what we're doing with collaborators in Austria who have also made some important findings in this area is actually to try to set up an international prevention study for BRCA1 mutation carriers. And these healthy women who haven't had breast cancer will be randomized 50-50 to either receive this drug denosumab to switch off that pathway or a placebo every 6 months with a single injection under the skin to see whether this hypothesis will actually play out. And the reason, of course, for doing it as a randomized study is that's the only scientific way of ultimately knowing whether or not this is going to work and potentially be important for these women, but more importantly, I think, for the next generation of women, because you need to have a black and white answer. And of course, anytime you give a drug to a patient, you can potentially do harm. So there's an important case to be made for having a placebo, because part of the study will be monitoring whether or not there are side effects to this. So I'd like to quickly change tack to another area that we're pursuing. And we've developed a range of models in our laboratory where we can take tumours from women affected by breast cancer. So at their diagnosis or at their surgery, they donate to our tissue bank some of their breast tissue, which is in excess of requirements for diagnostic purposes. And we're able to now, using the technologies we use to study human stem cells, we can actually grow these tumours in our laboratory animals. These are immunocompromised or immunodeficient mice in which you can grow human tissue. And that actually sets up a perfect scenario where you could study the behaviour of that tumour if you were to treat the mice with drugs just like you would treat a human, because these tumours actually very much recapitulate what would be happening in the patient. And so one of the areas that we've been particularly interested in is to try to tackle an area in breast cancer that really hasn't been a big area of focus up until now. And one of the things that a cancer cell obviously has to do is to know how to divide and grow. And of course, there are a whole set of genes that are important for that. And estrogen, for example, can very much help with that. And it's this proliferation which a new class of drugs called the CDK4/6 inhibitors are targeting. But cancer cells also have to be very smart. They have to know not to die. And even in the face of chemotherapy or hormone therapies. So they develop a whole string of mechanisms to help keep themselves alive despite the effect, the normal programming of cell death that should be happening in these cells or in response to treatments. And so we've been studying a group of what are called pro-survival proteins, which belong to the BCL2 family. So BCL2 is the hallmark gene from this family that helps keep these cells alive. And so what we've been doing is looking at an inhibitor of BCL2. And in this case here, you can see when we treat these mice with tamoxifen, we do see some effect. This is what happens with placebo. The tumours just grow incredibly rapidly. With tamoxifen, we can affect the tumours. But if we use this combination therapy by switching off BCL2 and using tamoxifen, we can actually, in this particular model, we were able to eradicate the tumours. And we've looked at a few other models and we see a similar effect whereby these BCL2 inhibitors synergize with tamoxifen to make it more effective as an anti-cancer drug. So this has now gone to the clinic where we're doing an early phase study. The The drug that we're using is called venetoclax. It's been developed to treat leukemias and got very rapid approval by the FDA in the US because it showed such tremendous promise in chronic lymphocytic leukemia. So we're now asking the question, well, is this relevant for breast cancer? Because about 75% of breast cancers have high levels of this BCL2 protein. And these are the first 15 patients we treated on the dose escalation component, where we're trying to find the right dose, because we don't actually know what the right dose is to treat patients with. But this is called a waterfall plot, where if it falls downwards, that's good, because it means the tumour is getting smaller from baseline, which is along here. And you can see that even in this dose-finding study, we saw some very impressive responses with tumour shrinkage. And now we've entered the escalation phase. We've identified that the drug is safe in combination, and we're starting to look at efficacy. And this is just to give you a flavour of some of the things we're seeing, where this, for example, is a lady whose cancer relapsed after 4 years on Arimidex or anastrozole with bone and liver problems. And we treated her with the combination drug. And this is what's called a PET scan, where you can see the areas of hot activity from the tumour, and within 4 weeks we've seen a metabolic shutdown of the tumour growth in this patient. And we're beginning now to look at about 20 or 30 patients, and hopefully there'll be other studies to follow, because once you see that a drug is safe and you see that it's potentially effective, the next question is, well, can you confirm that by doing a randomized study? So is this just tamoxifen, or is this more than tamoxifen? So that's hopefully going to be the next generation of studies that we'll see. And so I'll leave it there. The work I've presented is obviously the culmination of many, many people's efforts over the last 20 years for our lab, and Jane Visveda and I co-head the lab as a sort of nerdy husband and wife team of scientists. So I'll leave it there for questions. Thanks very much.
Right, we're running slightly late, but we do have time for 1 or 2 questions, if there are some.
Yes, you talked about the tamoxifen. What about letrozole?
Letrozole. So in the particular way we do the mouse models, we're not able to test letrozole or Arimidex or exemestane, which are the other belong to a family of aromatase inhibitors. It's just the way that the models are set up. But we would expect that it would behave the same way with letrozole. It's just there are quirks to the way we did the modelling. We have looked at another drug, which I'm not sure if it's available in New Zealand, called fulvestrant or Faslodex. It has the same combo effect. So we're thinking that this is a generic observation for women with hormone receptor positive cancer.
Any further questions? Marvellous. Thank you very
Thank you.
much,
Thanks.
Professor Lindeman.